Hello friends, welcome to BMH Learning. In this video, we are going to discuss about fat analysis by Mojonnier method. Introduction. During handling and processing of milk and dairy products, they are required to go through certain analytical controls in order to guarantee their composition and quality. The quality and the composition of milk and its products is mainly estimated by fat analysis, where we check the quality and quantity of fat a particular milk sample is having. Fat content determination is of immense significance because fat quality affects the price paid per liter of milk. It's very helpful in knowing if a sample of milk, or you can say other dairy products, comply with established legal values. Moreover, fat analysis becomes necessary in order to classify the milk for the preparation of its various derivatives. So, for estimation and analysis of fat, there is a method called Mojonnier method. It's a kind of gravimetric method which makes use of organic solvents for extraction of fat. Once the fat is extracted, the solvent is then evaporated and we are left with fat. The fat which is left after solvent evaporation is allowed to dry, and the fat content is determined by weighing the dried fat extract. Procedure. It's divided into 3 extractions. First extraction. Here is our milk sample added in special type of container designed for fat analysis. It's a Mojonnier tube. So 1.5 ml of ammonium is added. When we add the ammonium, the color changes to pink. Then we add 10 ml of ethanol. Next chemical is diethyl ether. So we add 25 ml of diethyl ether into the Mojonnier tube. The contents are then gently mixed by rocking over and back. We keep it for a few minutes. The last chemical we add for the first extraction is petroleum spirit. 25 ml of petroleum spirit is added. Then we allow it to stand for 30 minutes. Now you can see there are 2 layers. The upper transparent layer is of ethanol. So we decant the ethanol into a dish for evaporation. We are left behind with pink component, which is some protein. So in the evaporating dish, we have our first sample and we proceed to our second extraction. In the second extraction, the chemical composition we need to add is changed. The experiment is repeated and the ethanol is allowed to evaporate again. In the third extraction, the chemicals are added in this volume. So once 3 extractions are done, and after all the ethanol is evaporated, we get this. Now there are actually 3 samples. First is high fat milk sample. Second is light fat milk sample. And the third one is skimmed milk, which we are using as control. We can clearly differentiate the 3. In the first, the dried fat content is more. The second one has less dried fat, but the skimmed milk sample shows nothing. Finally, we put these 3 dishes in incubator set at 100 degrees centigrade in order to evaporate if anything left behind other than fat. And the amount of fat we get after incubation is weighed in an analytical balance. This was all. Thanks for watching.